So to represent this, you can see here from left to right, so we're looking at the sapling, which conducts water. From left to right, we have the increase in drought stress, and so decline in water potential. And as drought increases, more vessels become gas-filled and not water-filled anymore, and become non-functional. And once we reach a certain threshold, the plant will die. But plants have evolved different strategies to avoid this situation. So one of the main characteristics we can measure, that we call traits, is the ability of trees to tolerate xylem embolism. So to measure this, to quantify this, we build what we call vulnerability curves, which describe the percentage loss of hydraulic conductivity as a function of xylem water potential, which is the measure of drought stress in the tissues. So we typically have these types of curves, and here you have over 20 species, tree species from French Guiana. So on the right side, the curves most on the right side of the graph represent more vulnerable species. So the xylem pressure that causes 50% loss in hydraulic conductivity is quite close to zero. Let's say -2 would be -2 megapascals, which is a measure of pressure. It's quite vulnerable species. And as we move to the left here, we have an increase in resistance. So we're going to use this metric, what we call psi 50, which is the point where plants lose 50% loss in conductivity. So we see here that we have a strong interspecific variation in this forest, and this is also true in other tropical forests around the world. So as I said, once plants reach critical levels of loss in conductivity, they will die. But they have different adaptations to avoid this situation. So one of the first mechanisms in place is when plants experience a decrease in water potential during droughts, they're going to lose leaf turgor, and this will cause stomata to shut. So stomata are these small pores that exist on the leaves. And so you see here images of stomata at different scales. And on the bottom line, with the white arrows, are pointed out guard cells. And so when guard cells lose turgor, the stomata will progressively close. And this will drastically limit water losses. But even after stomatal closure, plants will continue to lose water through what we call residual water losses. So through leaves, through leaky stomata that didn't close well, or through cuticles, which is the leaf surface. So for leaf water losses, we call it G-min. Plants will also lose water through the bark, through lenticels, which are small holes in the bark. We've already observed this with our own eyes. What's important to see here on the graph below is that E-min, which is total transpiration from leaf and bark, is really uneven. Because in a tree, the leaf surface is tremendously higher than the bark surface. And so the amount of water lost from leaves is a lot higher than the amount of water lost through bark. So why is this important? Well, you may know that some trees lose their leaves during the dry season, for instance. So this is through a mechanism that we call vulnerability segmentation. Basically, it's when leaves are more vulnerable to embolism than the stem. So for example, on the right here, on the graph, you see a vulnerability curve for a leaf in blue and for a stem in red. And here we have a very strong difference between leaf and stem vulnerability. So let's say if you look at the x-axis, around -6 megapascals, the leaf will have lost almost 100% of hydraulic conductivity and will be shed, it will fall. But in the stem, it's still fully functional. There's no loss of conductivity at minus 6 megapascals. So at this point, leaves will be disconnected from the stem, and so they won't lose water anymore through the leaf. So this will really drastically reduce water losses because they will be only driven by the bark. So all these mechanisms, they interact to define what we call desiccation time. You see here on this graph, it's the time between stomatal closure, represented by turgor loss points, and the water potential causing tree death. So there are several phases during dehydration. The first one is when water losses are dominated by stomatal conductance, which is represented in blue at the top of the graph here. After stomatal closure, water losses are dominated by minimum leaf conductance. And when leaves become embolized and fall, then water losses will be determined by G bark on the right side. And when we reach high levels of stem embolism, which is the red curve, we have a progressive increase in tree mortality. So this is starting to be quite well described, how this works across species. But what we don't know much is how it can vary within a species, and do these characteristics change with the environment? This is what we call phenotypic plasticity, which is the ability to shift trait value with the environment or any other factor. And this might influence tree survival. So we have a quite limited knowledge of phenotypic plasticity, which is restricted to only a few intensively studied species. So for example here, pine species or poplar, which are very important trees in temperate regions. So on the left here, you see vulnerability curves for pine branches in different populations. The blue curve describes the vulnerability curve of well-watered trees, and the red curve of water-limited trees. And you see that they overlap quite well, and this means that there's no plasticity inside vulnerability in the species. However, for poplar on the right, you see that with the water limitation, the vulnerability, so in red here, it's leaves in dashed lines and stems in soft lines. And so with water limitation, these trees will shift the vulnerability to embolism towards more negative water potentials. This means they become less vulnerable, but there is plasticity. So it seems that it's quite species dependent. However, there's a considerable knowledge gap for tropical species and mountain species in particular. So this is the topic of the study that we realized in Rwanda a couple months ago. So this was realized within the Rwanda Tree project. Maybe some of you are familiar with it, some of you maybe not. So I'm going to present the overall project, sort of, so you have an overview. So it aims at studying tree growth and mortality in relation to climate, and to analyze changes in community composition. In this project, we also study tree physiological and structural responses to climate change. So this is the topic of study I work in. And one of the overall goals is to inform on tree plantations across Rwanda using native tree species. And this is done in collaboration with RFA and Drought. So the project consists in mixed plantations of trees at 3 sites in Rwanda along an elevation gradient. And we call this a space for time approach. So I'll explain a bit more after. So we have 20 tree species native to Rwanda. Some are early successional, some are late successional, and half of them originate from mountain forests, close to Nyungwe Forest. And half of them originate from Lake Victoria transitional forest at a bit lower altitude. So all these species were planted at 3 sites, one in Sigira, close to Nyungwe, which is at 2,400 meters elevation, which is the high elevation site. They were also planted in Rubona, which is the mid elevation site, close to Huye, and in Makera, the east, in the eastern province, which is the low elevation site. So around these sites, we have different forest types, mountain rainforest in Sigira, transitional forest in Hibona, and bushland and thickets close to Makere. All these sites present different temperatures, precipitations, and VPD, which is vapor pressure deficit, a measure of air dryness. Basically, on the west side, the high side, it's wetter, it's cooler, and it's not as dry. And on the east side, it's much drier. So this is called a space for time approach, because planting trees from, for example, the mountain forest at lower elevation, it mimics climate change. For example, in Verbona, it's 5 degrees hotter than in Sigira, and so it's in line with the projections of climate for the future. So this allows us to study species as if we were in the future. So this is an example of one plot design in Sigira. So we have in each site, there are 18 subplots consisting in different treatments. We have different irrigation treatments or water withholding treatments to mimic the different sites, and also nutrient addition to also mimic nutrient availability in the sites. And so each small square here that you see has 100 trees. So a few pictures so you can realize. So this was at the beginning of the project to set up the irrigation treatment. The colleagues in the project, they built rainout shelters to divert rainfall from some plots, so which mimic dry conditions. And here you have a view from above of the Rubona site after 2 years. So you can see on some plots the rainout shelters. Yeah. This is a picture from the Makera site after 2 years after planting. And now it's 5 years, so these trees are much, much taller. So they're now small forests. So in this study, I took advantage of this design at the high sites, Sigira and Rubona, mid-elevation sites. And we wanted to answer 2 main questions. Do hydraulic traits show a catastrophic response to hotter and drier growing conditions in Afro-mountain tree species? And will this trait plasticity, if it exists, will it minimize tree vulnerability to climate change? So for this, we studied 4 species, including 3 species from the mountain forest and 1 species from the transitional forest. 2 of these species are early successional, and 2 are late successional. We measured several traits, including vulnerability to leaf and stem embolism, building these vulnerability curves that you saw before. We measured leaf turgor loss point, which is a measure of when stomata close during drought, and residual water losses from leaf, G-min, and bark, G-bark. And to get this net effect of trait plasticity and values, we used a branch-level hydraulic model to include all the observation. And this allowed to calculate desiccation time, which we call T-crit. A point on the methods. So to measure xylem vulnerability to embolism, we relied on an optical method. So basically, we harvested branches in the field, brought them back to the lab in large plastic bags to avoid them losing water. And then we attached small cameras, you see here in red on the scheme on the right. We call cavity cams, and they take pictures every 5 minutes during branch dehydration. And from image analysis, we can identify embolism events in the leaves and in the stem, and then reconstruct the dynamics of embolism and hence loss in hydraulic conductivity. And to measure water potential, we attached a psychrometer to the stem, presented here with this small rectangle, and this measures water potentials continuously. So now let's see some curves. So here show these curves on the left, it's one species. On the right, it's another species. You might be familiar with them. On the top panels, it's results from the high site in Sigira. At the bottom panels, results from Rubona. So let's start here on the left with Macaranga. So the red curve represents the leaf, and the blue curve the branch. So you see here that from the high site to the mid-elevation sites, you have a shift towards the left, which means that there is plasticity in these traits for leaves and for stems, so they could adjust a bit of this trait towards more resistant values. On the right panel for Syzygium, you see a bit of the same pattern, but the plasticity for leaves was stronger than for stems. So you see that the distance between the blue curve and the red curve is almost non-existent in the low site of the bone. So the vulnerability segmentation pattern is sort of constant. On the contrary, on the left side, Macaranga sort of conserved this distance between the leaves and stems. So these species are slightly different in vulnerability. Macaranga seems a bit more vulnerable than Syzygium. I will jump to the two other species, which are much more resistant. On the left, you see Forea, which is a lot more resistant, but also we see a tremendous segmentation pattern, where leaves are much more vulnerable than stems. You can also see that we also have a shift to more resistant values. You see that the vulnerability curve is shifted towards the left for leaves and for stems. And finally, on the right panels for Croton, which is the only, let's say, lower altitude species, there's only one curve for the branch, because I couldn't measure leaves, because they were a bit too thick and hairy. But what's special here is that you see also a shift in the vulnerability curve, but in the other way compared to other species. So here at lower elevation, where it's hotter and drier, Croton branches are actually more vulnerable to embolism. So I'm not sure why this is. It doesn't make sense intuitively. But it may be due to the fact that Croton grows much better at lower elevation. And we often observe a compromise between growth and survival. So maybe higher growth at low elevation means that they're a bit less resistant to drought. Anyways, this was the most resistant species compared to the three other ones. So maybe we don't really need to be so resistant. Now if we look at residual water losses, so on the left panel here, we have leaf residual water losses, G-min, and on the right, bark residual water losses. So we see generally a similar pattern. So the blue boxes are for Sigira, yellow boxes are for Rubona, and so you have the four species represented here on these box plots. And so the general trend is towards a decrease in minimum conductance of leaf and bark at the low elevation sites. So with drier and warmer conditions, plants limit water losses through these tissues. So we have a plasticity in this trait as well. So now we're interested in seeing the net effect of all these trait variations on desiccation time. So we used a desiccation time model, which I simplified here. So it has two main components. One is the time from stomatal closure to leaf hydraulic failure, when leaves will be disconnected from the stem and drop. And the second component is the time from leaf to stem hydraulic failure. And so from this, we can calculate desiccation time, which is T crit. So first result here is when we consider trait plasticity, but we don't consider the change in the environment. So in blue here, you have desiccation time for trees in Sigira, and yellow for Rubona. So you see that for most species, trait plasticity actually increases T crit. So the yellow bar is higher than the blue bar. This was not the case for Syzygium, SGU here. probably because there was a cancellation of its segmentation pattern. But the net trend is that we have an increase in T-crit with trait plasticity. But now, if you think of the different climate conditions in which these trees grow, um, you can use these climate data to compute T-crits in a more refined way. So I used vapor pressure deficit, the measure of air dryness, which is twice as high in Rubona than in Sigira. So it's a lot drier in Rubona. Integrating this effect of climate, you see that T-crit is almost always lower in Rubona than in Sigira, which means that even though trait plasticity increases T-crit, it may not compensate for hotter and drier conditions. So we only have partial acclimation of vulnerability to drought. So what can we retain from this study? We showed strong interspecific differences across species. Mountain species appear to be more vulnerable than the transitional one. We also showed that hydraulic traits show a plastic response to warmer and drier conditions. But we saw that there was contrasted plasticity across traits and species. They don't all respond the same. And for me, I think, which is the most important, is that the magnitude of hydraulic trait plasticity may not minimize Afro-mountain tree species vulnerability in a changing climate. So in this project, to go further on our mechanistic understanding of why communities change in composition, we'd like to include other traits, such as leaf phenology, which is when do trees drop their leaves during the season, rooting depth, trees that can go reach water at in deep soil levels. They can probably sustain hydraulic transport longer during drought. We also want to include tree allometry, which is tree dimensions, so we can better estimate how much water is lost during drought at the whole tree level. So we can model whole trees and not only branches, as I did here. And this could serve to parameterize dynamic global vegetation models, which are more large-scale models, and they're able to have an idea of how communities change at a regional scale and inform us on carbon fluxes at an ecosystem level. So with this, I'd like to thank you for your attention. And if you want to know more about the Love Tree project, you can go on the website of the project. Thank you.
Yeah, thank you very much. Yeah, it seems interesting. I want to see if any one of us has a question or comment.
Well, feel free.
Yeah. And your question is any comments? This is your time.
Did you have comments on your personal experience of what you see around in Rwanda?
I don't see any question.
That's
Hello.
fine.
Ah,
Yes, hello.
yes.
Hello.
Hello.
Uh, thank you, the presenter, the speaker, Camille. It was very, very interesting.
Thank you.
In fact, I did not get well whether from these different types elevations across the country. You know, you have in the south the high elevation, then up to the eastern in Makela. So I would like to know whether you are dealing with the indigenous species or you are also mixed up with exotic species, tree species in that particular areas from different tree areas.
Okay. Well, this project was done in collaboration with RAB and RFA, and one of the goals is to increase the part of native tree species from Rwanda in tree plantations, which are currently dominated by eucalyptus species. So they grow very well. They provide good wood, but they also cause some environmental problems related to soil erosion, water quality, and biodiversity. And so this is why RFA and RAB, they want to include more native species. So in this project, the tree species used are only from Rwanda, but they come from different locations. Some are